STAR stands for Scientific Training in Antimicrobial Research Strategies, and its main purpose is to provide scientific training to 9 PhD fellows and 1 postdoctoral scientist in antimicrobial discovery and development. The project has 2 main sub-programs, one on target-based approach to developing new antibacterial drugs and one on anti-malarial drugs. Both themes are addressing the problem of drug resistance. Drug resistance in bacterial pathogens continues to make headline news, and even our last resort drugs for multi-resistant pathogens seem increasingly fallible. The malaria parasite infects more than 200 million persons every year and causes over 1 million deaths annually. Malaria drug resistance is spreading now across the globe, and it's linked to the increased incidence of morbidity and mortality. Here we present the results of our target-based discovery and development program for new antimalarials.
Malaria remains one of the most infectious diseases worldwide. Every year, almost 1 million people die of malaria, especially children and pregnant women. The most severe form of the human malaria is caused by a parasite called Plasmodium falciparum. Despite the fact that there are many anti-malarial drugs on the market, such as chloroquine and artemisinin, resistance to these treatments is an increasing problem. In fact, the first eradication program organized by the World Health Organization in 1957 has failed. There is therefore an urgent need of effective treatments against malaria. We are working on a new anti-malarial target that is essential for replication of malaria in humans. The cycle of malaria starts with a mosquito bite. This is when the parasites get in the bloodstream and infect red blood cells. Here they replicate, rupture the red blood cell and get released in the blood, where they can infect new cells. This leads to the major symptoms of the malaria disease. One of the proteins essential for the escape of the parasite from the red blood cell is a serine protease called PF-1. PF-1 cleaves other proteins which are necessary for the rupture of the red blood cell membrane. By inhibiting PF-1, the parasite cycle in humans would be stopped. To find inhibitors of PF-1, the National Institute for Medical Research in London is working together with Inibox in Oxford and the Latvian Institute of Organic Synthesis in Riga.
Inibox is a spin-off company from the University of Oxford. It has a long-standing expertise in virtual screening and has developed a huge database which contains more than 6 million compounds which are drug-like and commercially available. This database is used in high-throughput virtual screenings on targets such as PF-1. The catalytic domain of the PF-1 structure looks like this. It's a large globular protein which contains all the important structural features characteristics of subtelases. This includes 8 alpha helices, currently shown in red, several beta sheets, shown in yellow, and loop regions. The binding site, currently highlighted, is a shallow groove where peptide loops of other proteins can bind. Let's have a closer look to the binding site. The substrate binding site forms a cleft that allows a perfect fit to the substrate structures. In addition, a large number of stabilizing hydrogen bonds are formed between the peptide and the structure. They are currently shown as dashed lines. The S1 pocket harbors usually polar amino acids such as the present glutamine. The S2 pocket is very small and allows only small amino acids such as alanine and glycine residues. S3 does not really show preference as the side chain points away from the binding site towards the solvent, and S4 is highly hydrophobic. The prime site residues have a preference for hydroxyl containing and acidic residues, which is unique for this class of proteins. This also explains the unusual requirement that PF-1 requires the interaction of both prime and non-prime site residues of the substrates. PF-1 is a serine protease, which means that its function is to cleave other protein structures. The basis for these reactions are 3 residues which form the so-called catalytic triad. In the case of PF-1, this is formed by a serine residue, a histidine and an aspartate. Together, they catalyze the cleavage of the peptide sequence. As mentioned before, the goal of the study is to find small molecules which block the function of PF-1 and can be used as new malaria drugs. One approach to find such molecules is to test in silico the complementarity of thousands of compounds. Those which are predicted to bind the best are then synthesized in the chemical laboratory in Riga and afterwards experimentally tested in the National Institute for Medical Research in London.
To make the predicted inhibitors of PF-1, different synthetic schemes were developed and tested. The Latvian Institute of Organic Synthesis has extensive experience in drug development. Over the last 60 years, it has been involved in the development of several drugs on the market.
More than 700 reactions have been executed for the synthesis of PF-1 inhibitors. Reactions are monitored consistently using a method called a thin layer chromatography. To ensure successful synthesis, all compounds are analyzed by complementary analytical techniques in mass spectroscopy, liquid chromatography, and nuclear magnetic resonance spectroscopy. The final compounds are sent to the National Institute for Medical Research in London for biological testing.
The compounds are sent to the National Institute for Medical Research in London. This institute has contributed to several major biomedical advances, such as the discovery of human influenza virus. Here, our group is studying the role of PF-1 in malaria and testing potential inhibitors. For this, compounds from Riga are solubilized and tested on the recombinant protease PF-1 as well as on the parasite in human blood. In 2012, the STARS-Marie Curie collaboration published a set of new PF-1 inhibitors. This project has provided promising inhibitors for the development of new anti-malarial drugs.